A still pond at dawn. The water is flat as glass, and the surface looks like a sheet of emerald felt, so complete that you could cross it without breaking the green. Up close, it isn't a single film. It's a carpet made of millions of specks, each smaller than a grain of sand, each a complete little flowering plant. This is not algae. This is Wolffia, duckweed, one of the smallest flowering plants on Earth. And some strains of it can double their biomass roughly every 29 to 48 hours. One day to the next, 2 days later, 4 days later, exponential growth that looks almost like magic. If you want more science-backed explorations of nature's overlooked solutions, subscribe and stay with us. This story starts at the water's edge, but what follows stretches from backyard buckets to wastewater treatment, from Southeast Asian food markets to experiments that imagine feeding astronauts in orbit. So what exactly is Wolffia? Picture a tiny green dot, usually under 1 millimeter, floating free at the surface. It has no obvious leaves or roots in the way we typically imagine plants. It reproduces mostly by budding, creating clones that split off and expand outward. That simple mode of reproduction is why, under favorable conditions, light, nutrients, calm water, it can double in less than 2 days. Imagine compounding interest, but in living biomass, a single patch can rapidly become a mat thick enough to scoop with your hands. The biological curiosity is only the start. Some Wolffia strains, particularly Wolffia globosa, have been measured at up to roughly 45% protein by dry weight. To put that in context, soy is often cited around the mid-30s% range by dry weight. Quinoa is much lower. Many meats on a dry weight basis fall in a different ballpark. Laboratory analyses show that Wolffia contains all 9 essential amino acids in proportions that meet or approach the World Health Organization's recommendations, and measured protein digestibility in at least one clinical trial reached about 89%, comparable to high-quality animal proteins. That trial, conducted by researchers at Ben Gurion University and published as a clinical study, involved adult male participants and compared Wolffia-derived products to other protein sources, providing a controlled context for bioavailability and safety assessments. There is another surprising twist. Some studies have detected biologically active vitamin B12 within Wolffia tissue. The most rigorous confirmations use liquid chromatography mass spectrometry methods, LC-MS, to distinguish true B12 from inactive analogs. The likely explanation is not that the plant synthesizes B12 itself, but that bacterial endosymbionts or associated microbiota within the plant tissue contribute or carry the vitamin. That discovery matters because B12 is typically absent from plant foods and is a central challenge in plant-based diets. All of this produces an obvious question. If a speck of pond green can deliver protein and even B12, why hasn't it become a mainstream crop? The answer is less about conspiracy and more about structural misalignment. The 20th century invention of the Haber-Bosch process, industrial nitrogen fixation, made synthetic fertilizer cheap and abundant. That enabled vast expansions of soil-based, high-yield monocultures that fit an industrial supply chain. Big fields, mechanized sowing and harvesting, seed companies, fertilizer companies, bulk transport. A tiny, freely reproducing aquatic plant that thrives in decentralized water basins doesn't slot neatly into those systems. Research funding, subsidy frameworks, and commercial investment have tended to reward crops and technologies that scale within existing infrastructures. So for decades, attention and capital flowed elsewhere, leaving duckweed largely in the margins despite its remarkable biology. That marginalization had real consequences for research trajectories. Early interest in aquatic plants did produce experiments, some by agricultural agencies, some by space program researchers, but results were patchy and often applied to different genera. Not all duckweeds are identical. Some species or strains produce compounds like oxalates at levels that raise safety questions, and laboratory results from one species should not be generalized across the family. NASA's early work, for example, often focused on Lemna species for closed-loop life support studies. Later comparisons showed species-level differences in growth, nutritional composition, and secondary metabolites, which complicated the narrative that duckweed is uniformly ready for human diets. In short, a few misapplied or poorly generalized experiments seeded skepticism that stuck longer than it should have. Still, the empirical record for certain Wolffia strains has been building. Laboratory growth curves and cultivation trials show remarkable doubling rates. Some Wolffia microscopica strains even reproduce faster than the average 29-48 hour window under optimal conditions. Yield estimates reported in plant science literature suggest theoretical dry matter protein outputs that are orders of magnitude higher per unit area than many conventional crops, but those figures depend heavily on controlled inputs like nutrient concentration, light intensity, temperature, and species. Phytoremediation studies add another layer. Duckweeds, including Wolffia, have been reported to remove large proportions of nitrogen and phosphorus from contaminated water. Some trials reporting removal rates on the order of up to 99% for nitrogen and up to 88% for phosphorus under particular laboratory or field conditions. That means a single cultivation system might simultaneously treat wastewater and produce biomass suitable for processing into food or feed. But again, species choice and water quality are decisive. There are practical reasons these findings matter beyond academic interest. Space agriculture programs have been attracted to Wolffia for decades because it grows on water, has a short turnover time, and packs lean bioavailable protein into tiny biomass. Simulated microgravity experiments reported in recent years suggest that certain Wolffia strains maintain productivity under altered gravity conditions, and researchers have run feasibility calculations indicating that a few hundred grams of fresh Wolffia could supply an astronaut's daily recommended protein intake, depending on moisture content, processing, and exact strain composition. Those calculations are promising because they combine high growth rate with compact volume, which are both critical parameters for closed-loop life support systems where resupply is limited. Again, the caveat is that these are study-based estimates, not operational guarantees. Engineering a reliable, palatable system for long-duration missions still requires further testing. Commercial pathways are beginning to move from lab benches to markets. An Israeli company has commercialized a cultivated Wolffia product under the name Mankai and partnered with researchers to publish clinical and nutritional studies, demonstrating safety and bioavailability in controlled settings. The European Food Safety Authority has evaluated certain novel food dossiers and moved toward approvals in recent years, opening regulatory pathways for consumer markets. Peer-reviewed work, including a 2024 scientific reports paper, has strengthened the evidence base in contexts that include simulated space conditions and scaled cultivation studies. Taken together, these developments signal that the science is progressing from curiosity to cautious commercialization. But practical caution is essential. Not every duckweed patch is fit to eat. Species and strains differ. Environmental contaminants concentrate in aquatic plants, and simple harvesting without processing can leave behind pathogens or unwanted chemical residues depending on water source. Some genera accumulate oxalates or other anti-nutrients at levels that require processing to reduce. Therefore, safe adoption demands species identification, controlled cultivation in clean water, and processing steps that may include rinsing, heat treatment, or incorporation into formulated products. When studies report high protein percentages or B12 detection, they do so for specific strains under tested conditions. Those results do not automatically transfer to wild harvested samples from an urban pond. For people curious about hands-on cultivation at a small scale, there are practical high-level options that prioritize safety. Start with a verified culture from a reputable source rather than scooping pond water. Identify the species and strain if possible. Use potable water or well-characterized nutrient solutions. Maintain basic hygiene around the growing vessel to keep out contaminants. And avoid cultivating in water exposed to industrial runoff, agricultural effluent, or unknown sewage. Light needs are modest but consistent. Calm water and a shallow basin encourage surface mats. Because Wolffia grows by vegetative cloning, contamination with unwanted species can be a management challenge. Regular inspection and, if needed, controlled culling of unwanted plants is important. These are cultivation principles, not recipes, and anyone considering producing food should consult up-to-date protocols and local regulations. Beyond production, there are broader social and environmental implications. In parts of Southeast Asia, Thailand, Laos, Myanmar, communities have long harvested and consumed duckweed variants known as Kai Nam or Ka Nam as traditional foods. These cultural practices provide recipes, processing methods, and culinary contexts that modern markets could learn from rather than dismiss. In humanitarian contexts, degenerated soils and disrupted supply chains make decentralized, water-based protein production an intriguing possibility. Imagine rooftop tanks, community basins, or integrated wastewater treatment gardens that turn pollution into edible biomass, if done with proper technical oversight. That's the dual benefit logic: food plus remediation. At the policy and industry level, the story of Wolfia highlights how research priorities follow incentives. Cheap synthetic fertilizers and mechanized agriculture built a dominant system optimized for certain plant forms. That optimization leaves alternative biological strategies understudied. This isn't evidence of deliberate suppression. It's a case of institutional inertia and funding patterns that favor scalable, familiar models. The recent pivot, regulatory openings, private sector pilots, and renewed academic interest shows that science can correct earlier blind spots when incentives and evidence align. So what should we take away? First, the basic facts are striking. Certain Wolfia strains can reproduce extremely fast, doubling in roughly 29 to 48 hours, while achieving protein concentrations reported up to around 45% on a dry weight basis. Some clinical work shows strong protein digestibility scores in controlled trials, and LC-MS methods have detected bioactive B12 associated with plant tissue, likely via microbial partners. Second, these properties are species and strain-specific. Not all duckweeds are interchangeable. Third, the environmental benefits, nutrient removal and low land footprint, add to the nutritional case, making Wolfia a candidate for integrated solutions rather than a single magic bullet. If we view this plant as a tool rather than a panacea, there are sensible next steps for those who want to move from curiosity to action responsibly. Support and share scientific work that is transparent about species, methods, and limitations. For community groups, pilot small, controlled cultivations using verified strains and ensure food safety protocols. For policymakers and funders, consider allocating resources to crop diversity research and decentralized food systems that do not assume large land tracts as the only path. For consumers, be cautious about unverified wild harvest sources and look for products backed by peer-reviewed studies and regulatory checks. We began at a pond at dawn because the image matters. Tiny life, unassuming and green, covering the water like a secret. That secret, once illuminated by careful science and thoughtful stewardship, could become a practical piece of a more resilient, diversified food system. It's not a guaranteed revolution. It's an invitation to broaden our toolkit and to pair ancient culinary practices with modern safety and scaling research. If you want to dig deeper, the links in the description point to the relevant research papers, regulatory documents, and commercial resources mentioned here. Ben-Gurion's clinical work, LC-MS B12 studies, recent simulated microgravity research, and the regulatory reviews that have opened new doors. Share this video with someone who cares about food, environment, or space. The next time you walk by a pond and see that flawless green sheet, ask yourself, what else have we been overlooking that might feed us, clean our water, or keep astronauts fed on a long mission? More vaults from nature are waiting. The question is whether we'll look.